Thank you for joining us today. And I am honored today to introduce Professor Saheer Gharbia. Professor Gharbia is the head of Genomics Research Unit at the Public Health England. Professor Gharbia has over 25 years of experience in microbial taxonomy, deciphering pathogen diversity, and impact of microbial evolution on host pathogen interaction. Thank you, Professor Gharbia, for spending the time with us this morning. It is all yours.
Thank you, Clotilda. Hello, everyone. The webinar today is to share with you the lessons and the challenges that we and several members of the community, the scientific community that are working in the area of the human microbiome are attempting to explore and attempting to finalize in order to create a framework and a standard by which we can compare data, share information, and make informed decisions over what interventions and when and where they are required. The opinions and the presentation that I'm sharing with you today is my opinion and that of my colleagues that I'm collaborating with, and they do not and may not represent the opinions of the teams in Illumina, nor are they subject to any current process of FDA approval. When we talk about the human microbiome, we really talk about the component of cells that are within our body that are separate from the content of our chromosomes or content of the genes that are responsible and form the mitochondria. So it's really our other genome. And as you can see on this slide, we have 10 to the 12 human cells that form our single human genome with 3 gigabytes of sequencing, 3 gigabases of sequencing, and over 100,000 protein. While the mitochondria that also contribute to the functions of the human genome is formed out of 10 to the 14 mitochondria and 17 kilobases of sequencing, leading to the formation of about 13 protein. While the microbiome is the largest number of genes and chromosomes that are within our body, and there is 10 microbial cells for every copy of the human karyotype. There are almost 10 to the 3 or 10 to the 4 different species that form the human microbiome, and they are responsible for the introduction of 3 million genes at any time, and hence 3 million potential corresponding proteins. So as you can see, it's a very complex component of us. And over human evolution, they became part of our function, ability to adapt, response to diseases, and sustainability of good health in humans. There have been several human microbiome initiatives, and the recent set of initiatives over the last 10 years have been driven largely by the recent advances in sequencing, ability to produce highly computerized analytical system and bioinformatic tools in order to decipher variations and similarity between protein, gene sequences, and between a taxonomical group. And as a result of that, there has been an explosion of work in this area. Regardless of which direction and which group has done work, there has been an apparent need for a framework and for reference standards in order to take this comparative work further, and to be able to define whether we do have a core set of organisms that can form the human microbiome that we should be able to compare among the various organisms. The outcome from many of these bacteria was that there are two major bacterial divisions, the bacteroidetes and the firmicutes, that constitute about 90% of the organisms in the gut flora, and the 10% is shared among thousands of other organisms. So I'll try and focus today, while I'm discussing these areas, of impact on these two main groups. And it became also apparent from clustering and comparing that there is a direct impact of the microbial flora on the health of the intestinal tract. So they are the center of homeostasis between man and microflora. And although a lot of these initiatives are 10 to 15 years old, for the last 100 years, we have been, as scientists, exploring this area in further detail, with the focus initially being on the cultured flora and on phenotypic observation in order to characterize these organisms. And I must admit that now that we have moved into a lot of more of the genomic analysis and being able to compare the function and structure of these organisms, none of the new information have deviated from the framework that had already been established in this area. However, what is quite clear is that among the organisms that we can now identify either by direct sequencing or by culture or non-culture method or proteomic method, and any mechanism that we consider, we only know about 40% of the microbial genes that are present within these individuals, and we have only been able to assign bacterial names to about maximum 48% of the reads that we have characterized, which means that there is at least 50% of the microbial flora that we do have genetic signatures for, but we do not have a species allocation and naming process for them. And that will take a period of time in order to establish and in order to create a framework of how we can characterize them. It also became quite obvious that there is a shift and changes that do take place in the human gut microbiome. It's a dynamic environment. Age, nutrition, environment, and host physiology all affect the sum of these changes. So the main focus has been to try and characterize what constitutes the main component, what is the core genome. So before we go into more details of that and the challenges that have been resolved and the ones that do exist, I'd like to share with you the view that was presented by Whiteside et al. In 2015, which is what is a microbiota, what is a metagenome, and what is a microbiome. And therefore, when we characterize organism, are we talking on what is presented in slide A, the taxonomical entity that identify each and every component within the cell. And most of these have been identified or are compared based on 16S ribosomal RNA gene sequencing. So they are compared to one component within the chromosome that could be used for taxonomical allocation to define what are the content of the microbiota. When we undertake metagenome analysis, we characterize sequence reads that are identified against the chromosome, against the plasmids, against the phages, and against the various components and genes within the cells. While when we refer to the microbiome, it becomes a bigger challenge, but also a more interesting comparison, because it also covers the metabolites, the protein, and what is expressed by the organism. And in some cases within the microbiome, we are also characterizing some of these physiological and metabolic property of the host that are produced as a response to the presence of the organisms or as part of their interaction with the bacterial flora. So I wanted to define the three components and how I will refer to them in the next set of slides. So in this slide, based on 16S phylogeny and based on the work that has been done over the last 10 years, we now identified that in the microbiome of humans, the gut microbiome is predominantly bacteroides and firmicutes. They form about 80 to 90% of the flora, while proteobacteria and other organisms constitute about 10%. And therefore, there is need to be able to look at how do we create standards for characterization, for comparing sequence reads, and for referring to the core flora that will accommodate these two sets of characteristic group of organisms. With firmicutes, it covers the Clostridia, the Lactobacillus and related species, while the bacteroidetes covered from Bacteroideaceae all the way to other gram-negative anaerobic bacteria that are related to them. The proteobacteria, which is shown in the top of the slide, cover the Enterobacteriaceae and other gram-negative facultative anaerobes that are related to them. And it's just to give you a reference point for each one of these groups. Now, when we undertake 16S analysis, we need to be really aware of how we interpret the data and what are the challenges associated with it. So on this slide, what it is the 16S gene that we use to compare organism with vary in its number across the genome. Some organism may have only one copy, such as this purple D here. Another organism might have 3 copies of the 16S, 2 copies, etc. As you can see in the slide, we can have a whole set of organisms that vary in the number of the 16S loci within the genome. And just for comparison, in an analytical study by Lowe et al. In 2011, they've used in silico production of a mixture that has genomes with this varying number. And the percentage that is on the left-hand side to each one of them is saying, so if you have a bacterial lysis that contain 40% of organism D, 30% of organism C, 20% of B, and 10% of A. If you undertake targeted sequencing in order to amplify all of this mixture together, then you will get a variation in the bias of which ones are amplified and therefore which ones are red, and what is the estimated number of each one of these organisms in the final mixture or the final read output from whatever sequencing system you are using to sequence them. And if you look then on the bottom of the slide, you will find that organism D is underrepresented by 50%. So there's a reduction of 50% of the number of reads that are associated with this organism. And this indicates that using 16S analysis as a way you've estimated the abundance of flora without taking into consideration how many copies of the 16S loci on the genome of this organism can be misleading. So organism C was only 30%. However, after targeted sequencing and sequence data analysis, the indication is that it forms 45% and is the most abundant organism based on sequence data. So then there's a need for an accurate and a fast estimation, and that has to be linked to the number of loci. If any of the sequence data is going to be used as an estimate of the prevalence of an organism, its abundance, within the microbial flora. So that's both a challenge, but also a marker indicator that we need to ensure that we are aware of these variations, or that the sequence data that is being analyzed should be analyzed based on the presence and absence of organism rather than on the percentage, if we are not going to take into consideration the copy number of the 16S loci. The next challenge is, as I show on this slide, this is the standard familiar sequence in two-dimension of the 16S loci. And as you can see on the gene, there are areas that are constant and there are no variation between organisms, and there are regions that now we define them from V1 to V9 that are variable. And it's within this variability we can detect markers for various classes of bacteria, genera, and species. Now, the capacity of each one of these variable regions to provide a diagnostic or an identification marker for any group of organisms vary. And we need to be aware when selecting which variable regions are going to be sequenced and compared between organisms to the bias that each one of these areas may introduce on the sample, or which selective groups might be differentiated a lot easier than the other. And just for simplicity, if you look on this slide, starting from 100 to 1400, C1 is the constant region 1, V1, V2, and so on, and where they are and the sizes are under them. There is no single region that can differentiate among all bacteria. But by targeting multiple regions, then you would get a better resolution within a sample. And also to keep in mind that there are specific regions such as, for example, V1 is best at differentiating Staphylococcus aureus and from the rest of the Staphylococcus species, mainly the Coagulase negative that are a challenging group. So Staphylococci and other gram-positive cocci are a lot easier to discriminate among them using V1. While V2 and V3 are best for distinguishing most of bacterial species to the genus level. They don't necessarily give a high resolution to the species level, but by comparing V2 and V3, you get a clearer indication of which genera clusters are present within the sample. The exception for that is the Enterobacteriaceae, which are very hard to resolve beyond the genus level using, or even at the genus level using V2 and V3 region. And similarly, each area does have its own bias and its own region where it can cover. So therefore, it is quite important to get the most of taxonomical information out of a sequence technology. And as a result of that, being able to cover more than one variable region is vital. Regardless of which technology you are using, you do need to be able to combine sequence data concatenated from various amplicons within that. And for example, here I'm showing you on top, it's the Sanger-based standard sequencing of the CNS genome, and using either, for example, 454 Flex titanium, or using Illumina MySeq 300 parent read, you would be able to get a similar set of resolution that would allow you to cover this variable region if you target the through 3 sets of amplicons most of the variable region. And that would give an indication of the diversity within the sample and the taxonomical allocation more accurately. So, taking into at this stage what we've discussed, which is that 16S have been used as a way of creating a taxonomical framework, either by targeted sequencing or by any other methodology that you would use. It is important to be able to ensure that what is selected as the target, that you're aware of the taxonomical framework and which phylogenetic groups can be deciphered from that and which ones will be missed. In the next sets of data, I will share with you challenges, but also insight into the impact of the collection site, the reference data set that is used, and the metabolic property of the organisms on how we can compare sets of data and how this can affect the interpretation of the results. Okay, so just for reference, in my team and in our work, as to the various life methodology and see what we can infer from that set of information. There are 100 to 200 prevalent species that are available within the adult individual flora. If we need to compare those on a non-culture-based method to look at culturable and non-culturable, then there are a range of extraction methodologies. In our hand, we found that either direct extraction or a basic lysis, as that I've shown in this using mechanical method, gave us the highest level of resolution. Having said that, I will show you in the next set of slides that how you process the sample before lysis is more important than which methodology you plan to use. But what we have done is we have tried to use a front-end method in order to lyse or reduce the amount of human DNA, digest that out, sediment the microbial flora, and we used 6 different lysis methods, and they all give us similar or comparable DNA recovery of the microbial flora that are within these samples. And we've used that for both 16S libraries and for shotgun sequencing. Comparing our results, this is the number of the taxonomical entities that we have been able to resolve and the number of reads using 16S libraries versus shotgun libraries. And what we did in shotgun is sequence the whole sample and extract out of that the 16S comparable taxonomical data plus any unique gene sequence that are associated with certain taxa, species, or genera. And as you could see that you have an increase of 3-fold of resolution of the diversity within the sample using shotgun. So that's just to give you an indication of what is the limitation between both methods in the number of species or improving the diversity within any sample. These are the methods that we have used for comparing the sequence data, genomic data from row read, QC for those using these methodologies. I just wanted to share with you which software we have used for each step, from quality assurance down to separating the groups of organisms and putting the sequence data against the various reference sets. We've also, in parallel, have done functional analysis of the reads that we have, and I'll share with you later on what are the observations from that. So these are the methodology, both practical and computational processes, and the data that are shown in the next sets of slides are generating use these 2 combinations. We first looked at the impact of the extraction and freeze-flow cycling. So we took fecal samples that were tested, and we subjected them both as fresh samples to lysis directly or extraction directly from the fecal sample, and that's the first set of replicas that you have here. So these are two fresh samples where we have done direct 16S amplification from those for sequencing. The next two are ones where we've done the lysis that I've shown on the previous slide. We then, in parallel, took that same set because we had split that into replicas, and we froze the samples. As you can see in the next sets here, these are samples that have been frozen and thawed once, then frozen and thawed a second time, third time, fourth, and fifth. If you look in here, that is the abundance of the bacteria by phyla that we've been able to characterize from each one of these reads. The gray one on top represent the firmicutes, and the orange to brown represent the bacteroidetes. And then the bottom blue are the unassigned bacteria. The pinkish are the Enterobacteriaceae and other related organisms. And just roughly by looking at that, as you could see, that there is a great change in the percentage of some of the families, mainly the bacteroidetes, as a result of freeze thawing. So if fecal sample are taken from a patient and these are frozen for further analysis later on, the likelihood is that there will be a shift and a reduction in certain groups of organisms and their recovery upon whatever methodology or whatever lysis process you undertake following transfer of these samples. So the question that raises a variation here is, should the samples are either lyzed immediately after they are collected while they are fresh, or if they are going to be frozen, that are aware what organisms might be lost. From different patients that have been treated physically in the same manner. Let me just show you in more detail. So this is the effect of the extraction and freeze thawing, and they are divided by which group of organism. So these are the bacteroidetes, and this is 0, and this is after freeze thawing, the reduction in the abundance, and each one is one freezing thawing cycle. So in total, the organism have had a 53-fold change after 5 cycles. That leads, the bacteria are normally abundant at 32% within a sample. After the completion of 5, then the recovery we had was less than 5% abundant within each sample. What complicates the problem is then you start, because you are looking at the abundance within the reads, is it skews the data. So actinobacteria, for example, that would have been present or was present in this sample at less than 0.1% have increased by up to 2 folds as a result of the reduction or the loss of the bacteroidetes. Firmicutes remain around the same numbers, and therefore there was no impact of freeze thawing on the total abundance. And you see that clear in the literature in some publications where as a result of freeze thawing, the bacteroidetes are lost, and the abundance of Clostridia and Fusobacterium is increased within these sets of samples, and that's purely as a result of the lab processing rather than as the result of changes in the microbiota between patients. Similarly, if you look here, Proteobacteria are also affected, and a major reduction. So this point is you need to be aware of the impact of freeze thawing of samples on the preservation and number of sequences that are recovered, and hence the interpretation of the abundance of certain groups of bacteria within the sample. Since they certainly do not behave in a similar manner, you get a variation in the response to freeze thawing. Also, when we are inferring the content of the gut flora from either fecal samples or from any other samples, we need to be quite careful in the interpretation of that. And in this slide, I'm trying to show you, this is the total number of the taxonomical group, or what my principal taxonomical unit, whether it's a species, a strain, or a genus base, of the number that has been recovered from samples taken from hamsters. We collected cecal samples, rectal sample, and fecal samples, and we treated them all in the same way. We processed them under the same condition. And as you could see in here, these are the number of the total taxonomical groups that were recovered from the cecal samples. The number increases as you analyze rectal sample, and it explodes as you look at fecal sample. So by comparison of these three, it is apparent that the fecal samples represent the microbiota that has been collected, and almost comparing the fecal sample does not necessarily give you an indication of any changes that might affect the cecal or the rectal microbiota in a patient or in an animal or in any group of organisms macroorganisms that you're looking at. So it's to just point out that fecal samples do not give a direct representation of the content of the colon. They are likely to represent everything, perhaps from the oral flora all the way across the alimentary canal. But also, it is important to compare, when we compare sequences, is to compare them against the same data set. These are sequences that have been released from using the hamster method model from the fecal sample. And when we compare those against the human reference metagenome data, 76% are unknown species. And we cannot assign them to any taxonomical group, not even at the genera or the family level. Out of those, less than 30% we can assign them, with the majority are as lactobacilli and as bifidobacterium and ruminococcus. So comparing the microbiome of human to those of animals in the environment or animals that interact with, in order to try and assign modes of transmission or shared flora, become more challenging in the absence of specific microbiome data sets that can be used for the comparison of these organisms. So, with view of that now, what I will do now is share with you data that we have analyzed from human and human only, taking into consideration that we needed to compare fecal samples that were fresh, collected at the same time from groups within the same age group. And what we wanted to do is try and define if there was any in the fecal sample that any of the taxonomical group that may arise or may be part of the saliva or the mouth, or are we, when we refer to fecal sample, refer to everything down the alimentary canal from the stomach all the way down to the cecal, and what is excreted in the fecal sample. And with that definition, then we can compare what is a core flora. Are they similar? What are the ranges of diversity, and what are the percentage variation, using as a reference point how many copies of the 16S loci that I described earlier on, and its value and semi-quantitation. So we collected samples from the same patients. These are fecal sample, and these were saliva sample, and to represent the oral flora. And I'm pleased that it's still confirmed that the fecal samples have got sequencing, and observe taxonomical groups when we recover up to about 8,000 sequence per sample, that we can resolve them into two clusters. These are saliva samples and these are fecal samples. So from now on, I'll show you how that comparison. We said, okay, can we look and define what is a core microbiota in these samples? And we have assigned them to the various taxonomical groups. These are the Bacteroidetes, the Bacilli, the Fusobacteria, the Clostridia, and the Lactobacillus. These are fecal samples, so this is the average of these, which one of these groups that we found derived from the fecal sample, and this is the average in the saliva. And although some of the unassigned are similar, you will see that there is a reasonably clear separation between both. For example, the Bacteroidetes, 15% of the fecal flora, but we got less than 0.3% in the saliva sample. Apart from the Lactobacilli, which you would expect, which would be present in both, but by different taxonomical species allocation within the Lactobacilli. And Clostridia is the dominant within the fecal flora as a result of this variation. So there is an indication that if we can put some of the measures correctly in place, that we would be able to define components of the core microbiota, or at least that's what we thought looking at this first set of results. However, when we took that further, in here, as you can see, there is 100 fecal samples taken from patients from healthy individuals, all between the age of 40 and 60. And each one of these columns represent the various taxonomical groups that we found, and each group is given a special color. And the reason I'm showing you this is to show you that if you look across all of them, there is no apparent set of core taxonomical groups that are shared among all of these. We found that there was less than 20% that were common, and the percentage and the variation among them is too high to be able to define what would differentiate healthy patients from diseased samples. And that is a total odd to the samples that are taken from other sites, such as from the skin, from other parts of the body, and this is just an example of some of these. So therefore, when we look on the microbiota of the human gut, we have a far more complex set of data that we need to compare. We took this further by comparing pairs of twins that are premature twins, where one patient, one of the children, had necrotizing colitis, and the other one was healthy. We collected stools from these, and we characterized the flora among them, and we found we can get 25 to 30 taxonomical groups, with enterococci and staphylococci and anorococci being the main dominant one in premature babies. When we compare these further, we noticed that the taxonomical composition, every two pairs are next to each other. We've noticed that in babies, even diseased baby, their flora was more closely related to their twin than to any other premature baby that had the same disease. So the microbial flora seems to be stable and very, very similar, and there were minor variation between the set of twins compared to any other groups of patients. And this is more detailed in this slide. This is the complete flora taken two sets of babies, and each one of the species that we have characterized. The dark green is for the babies that had neck, and the light green are babies that were healthy and survived the disease. The same thing with the red and the pink. And as you could see from this, the sets of data that are collectively together are very similar between the identical twin as compared to between a common set of organisms that can characterize the progression of disease. This is just an example of what that gut microbiome looks like, and I'll leave it for you to look at it further in details later on in order to see it. But just to highlight that up until the last 2 years, there was an indication that Clostridium perfringens might be responsible for the progression of this disease. Now we recognize that that is not the case. We further analyzed this, and using shotgun sequencing, pulled the Clostridium perfringens sequences and recovered them from the premature babies to compare them. And what we did is we compared the toxin genes for Clostridium perfringens, and that is made out of 2 subunits. In the environment, when Clostridium perfringens is inactive, there is an insertion, a gap in here, and this gap makes it inactive. And we found that wherever we found Clostridium perfringens in these babies, this premature baby with disease, and at least 50% of them had passed away, that Clostridium perfringens had this gap, and as a result of it was a non-functional gene. So Clostridium perfringens cannot be the reason for the symptoms that are seen in these children. So by undertaking sequence analysis and comparison, we are now more aware that the variation that we detect have to be taken in reference to the patient, or the patient and their closest relatives share common physiological property, common food allergies, common conditions. And just to show you an example here, from the healthy babies that have not been given any antibiotics, and these are premature babies born at the same time in the same hospital, from the metagenome using shotgun sequencing, we have been able to analyze 39 antimicrobial resistant genes that were represented within their microbiota. And this is a heat map of these based on sequences. The one in red are the ones with the highest confidence and abundance, and the ones with orange are the ones where we had sequences between 200 to 400 base pair that reflects each one of these antibiotic resistance inferring genes. So therefore, the environment that the babies are in, or the environment that we all share, exposure to other antibiotics, and so do affect the composition of the microbiome, and also affect how many of these antimicrobial genes we tend to accumulate within our microbiome. And therefore, by analyzing these using microbiome, we can give an indication of which antibiotics may or may not be useful in treatment of ailments of the gastrointestinal flora. For reason of timing, I think what I will do is I will jump these 3 slides, forgive me, but to show you that on this slide here, these are the bacterial flora that were analyzed and compared in 2008 between various samples from the human gut microbiome. And these indicate exactly the same variation that I've showed you on our sets of slides from 2016. But what is genes, which means taken out of the gut flora, what are the genes that are there, and putting them against taxonomical groups that they refer to, and that's just showing here the various physiological and metabolic property of every group of organism. You will notice on this slide that there is a consistent set of genes that are very stable, and all the samples can show that the gut microbiome may not constitute similar core genomes, but it is likely to have core physiological and metabolic genes that collectively create functions that makes it quite effective. So if two organisms belong to the same cluster of bacteria, shown here, other proteobacteria, they are likely to have similar metabolic pathway for metabolism, for sugar production, for breaking some of the proteins in the gut flora that would favor having these organisms at a certain percentage within the gut flora. Similarly, for the Cyanobacter, the Bacilli, the Clostridia, and other group of organisms. And perhaps one of the challenges that we need to consider in the future is being able not to only identify the various bacteria, but to be able to, through shotgun sequencing, to characterize what are the core genes that are stable and are required within the gut flora. Because the bacteria numbers and entities vary with physiological properties, with age, and with nutrition, while the genes that we need in order to sustain a healthy gut microbiome have to be stable and be able to address what is required to keep hemostasis. I will leave you with that last point as a way of challenging where do we go from here once we create reference data set, once we decide what taxonomical frame we need to compare the samples to, where they are collected, and how we can ensure that we have a consistent comparative process as we go forward. I do hope that I have highlighted some of the consideration on the physical processing of samples, the collection of samples, what is a reference data set, and what are the comparative process that would allow us to move forward to characterizing a core flora or core gene as we move forward in this area. And I just finally would like to thank the organizers and my collaborators who have contributed to this intensive amount of work over the last 4 years. Thank you.
Thank you so much, Saheer. This was a great talk. I have a few questions online, but I'm going to start with my own. The one that startled me from the beginning, but you finished on, was do you think that we are going to be able to map out those 50% of the genes that are still unknown functions to us? And how do you see this happening in the future?
I think there are, that's a really good question, Clotilde. As we are sequencing more and more genomes, microbial genomes, we are now annotating them at the same time and characterizing the functional property of each one of these species. If we can now transfer the data sets from that for each one of these genes into reference data for microbiome analysis, then we would be able to assign not necessarily at this stage the exact gene, but the function of each one of these genes or which pathway they fall in by comparative analysis. So metagenomics cannot move forward without having full microbial genome sequencing happening at the same time for a large range of organisms within each one of these taxonomical groups. Does that help?
Yes. No, it really puts it in perspective. Let's go back to your freeze-dry cycle.
Yes.
So
freeze-sowing, not freeze-drying, sorry. It's freeze-sowing. It's when you take a sample from the patient, they automatically, at least in the hospitals here, these are then frozen at -20 or -80, and then transferred to a laboratory to process or to lyse the sample and sequence it.
Okay. So one of the questions was, what kind of standard did you use to actually compare all of them? an internal standard?
We have added to it a known number. We added and we added to a Fusobacterium and an Enterobacteriaceae and E. coli, known and quantified genomic sequence, and we added cells from both of them. So we used 4 standards in each one.
Okay, thanks. Another question is, you demonstrated nicely that the DNA extraction technique is a significant source of bias. Clearly, some methods work better than others for different phyla, but how do we assess the best method, and what is the metric?
I do agree with you. There is variation between the various methodology people use. In our hands, we felt that using a rapid lysis methodology with mechanical beating gave us the lowest level of bias.
Okay, nice. Another question is, have you looked through core flora and core bacteriophages infecting that core flora? And if there is no kind of core flora, but metabolite core couldn't be maintained by bacteriophage? Actually, that's a very good question.
Sorry, I didn't hear it clearly. Would you mind just repeating it?
Yes. So what they're trying to say is that is there a core? So when you're looking at the microbiome, not only there's the core flora, but there's also the core bacteriophage in there, right? So it's a balance between the two, and they're asking that actually how is the metabolite core really transforming one versus the other, and is there any way that we could really look at it as one big symbiosis in between the flora and the phages?
Well, the percentage of phages that in the microbiome has been a bit of a challenge. So it's one of the areas we are working at the moment, is to see if we can extract the phage component. Until now, our results indicate that it's an insignificant number for against the total reads using the current lysis methodology. So it would be good to hear from others who have worked on phages of what observation they had and which groups of phages they have been able to successfully analyze and isolate.
Yeah. Another question is, the effect of freeze thaw, so let's go back, is due to fragmentation of the 16S gene, differently from protecting the DNA in different bacteria and species. So in the same degree of freeze, would that effect be seen in shotgun metagenomics?
Yes, and it would be to the same level. So one of the criteria we've done is we had a minimum set of length reads, consecutive reads, and these were cleared using the QC criteria that I've shown on the informatics slides before we can analyze and compare those to each other. And anything below, we really had to discard, in the cases of 16S sequencing, at least 35% of the read, while for shotgun, we were discarding about 62% to 65% of the reads in order to ensure that fragmentation doesn't affect a bias in assembly and therefore inappropriate allocation to taxonomical group.
Okay. Another question is, do we know which bacteria have multiple copies of the 16S gene so we can compensate copy number in the analysis?
Yes, we do, and that's the slide I have given. On the bottom of that slide, the reference to the publication where this number of loci have been calculated. You can do them directly by referring back your species allocation to the full whole genome sequence of that organism, a model reference strain of it, and that will give you an indication. But that's a manual process, and you will have to do it for each one of the organisms. But by referring to the paper I've added at the bottom of the slide, you would be able to create that subset and perhaps build it into your analysis.
Okay. Another one that I had, at one point you said that the enterobacteria were very hard to resolve with doing 16S, but
yeah.
they're very important to the gut microbiome. So,
oh, no,
so
absolutely.
How would you
Yeah, 16S give you an indication on a familial level, and for some of the Enterobacteriaceae to the genus level, but not to the species. And it's only when you undertake shotgun sequence analysis you can reserve them to a more appropriate species allocation.
Oh, okay. So you have to do shotgun if you're looking deeply into Enterobacteria.
Yeah. If you wish to look deeply into the Enterobacteriaceae in order to differentiate Shigellas, for example, from Salmonella, you do need to be able to move to shotgun sequencing. So what I was trying to show is the limitation of each methodology, and therefore how would you interpret the data using the framework of the experimental design you had.
Okay. Another question actually that just come up, it says when you want to enable multiple types of research platforms, so for a single sample you want to do 16S, shotgun, phage, fungi, selection and culture, what do you think is the best approach of collection and storage at that point?
Fecal sample will give the most complex sample, but it's the least invasive in humans. So if it is in human, it will have to be fecal samples. In our view is that when the samples are collected, it is important to homogenize it and then split it into replicas immediately. Then these replicas are processed for the various lysis methodology. Ideally, lysis and extraction within 2 hours of collecting the samples provide the most consistent sets of yields. And once that is prepared, then the sample can be frozen rather than as a fresh sample that has just been collected.
Okay, and what would be the optimal amount that you would say? Do you think that out of a stool we would need 1 gram? Or so that you're sure to have?
No, I mean, I think you don't need as much as one gram, but it's we have collected samples that are up to about maximum 2 grams, but in any of these analysis, if they are homogenized, you don't need more than about 0.1 of a gram to 0.2 for each one of the replicas. And obviously, if you are looking for fungi or for phages that will be at a far less abundance within these samples, you may wish to go for a slightly larger sample. But ideally, homogenizing as soon as possible. And some colleagues' work I haven't presented because it was from different groups, suggested that if you remove the surface of the fecal samples and concentrate on the core within the matrix, the human matrix, you would get a better and more consistent recovery.
Perfect. Thank you so much, Saheer, for your time today. Thank you for giving such a great presentation for all of us.